Today, we're going to continue our discussion about the cell membrane, its structure, and its function by talking about cell signaling. I'm starting with a figure that you've seen before when we learned about the plasma membrane of cells. We talked about the fact that the plasma membrane is made of lipids, but also carbohydrates and proteins. And we previously learned that proteins can be peripherally or integral or even lipid anchored as far as their association with the membrane goes. And we learned about several roles that proteins play in transporting things into and out of the cell, right? So protein channels, protein transporters or carriers. But proteins also play another important role in the plasma membrane, and that is in signaling. So when we talk about cell signaling or a signal transduction pathway, we have a couple of elements that are always present. Right? There needs to be information received by the cell, and that is called a signal or a ligand. And the cell has to have a way to recognize that signal. We call that a receptor. Then there are a series of steps in which that cell converts the information in the signal bound to the receptor into a way that the cell can respond to it. And then there is something that happens at the end that's called a response. Right? So there's always reception, transduction, and response. And it's important to note a couple of things. First of all, cells require receptors in order to respond to a signal. Right? So a target cell cannot respond to a signal that it cannot sense if it doesn't have an appropriate receptor for that signal. Right? Some terminology. Again, I mentioned this previously, but a ligand is a molecule that binds to a receptor of another larger molecule. And that binding is very specific. Right? You don't want just any ligand activating any receptor. You want a very specific interaction between a ligand and its receptor so that the cell knows what it is responding to and what to do in response. Right? When the ligand binds to a receptor, it changes the receptor shape or binding abilities or interactions with other molecules, and we tend to refer to this as activation. So the receptor gets activated, at which point we have a signal transduction cascade followed by some sort of response. So if we look at a cell within a multicellular organism, there are lots of different types of signals that cell can be exposed to. And they all have names. Right? So an autocrine signal is a signal that affects the same cell that released that signal. Right? And you see that in the example at the top here. So the signal is responded to by the same cell that released it, so that same cell also needs to have a receptor for that signal. Paracrine signals diffuse a short distance and affect cells that are nearby each other. Right? So the signaling cell and the receiving cell are in close proximity to one another. The signal is released from one cell, it diffuses a short distance, and then is received by a nearby cell. Whereas juxtacrine signaling happens between physically contacting cells, right? Those cells must have a physical link, either be adjacent or connected by gap junctions, right? So the signaling cell must be touching the target cell, right? And so in this case, we're not releasing the signal into a nearby environment. It's actually passing directly from one cell type into another cell type. So this could be gap junctions, or this could be plasmodesmata connecting plant cells. And then finally, endocrine signaling is long distance signaling, right? It's where a cell releases a signal and that signal travels through the vasculature. So as far as animal cell signaling goes, you're talking about the bloodstream. Plant cells also have vasculature that transport fluids, right? So a signal could travel through those tubes as well. So we're talking about things like hormones, and that's where a cell type can release that information. It can transport long distances throughout the organism and be received by and responded to by a faraway cell or a distant cell. Right? So autocrine signaling, paracrine signaling, juxtacrine signaling, and endocrine signaling. So let's look at the receptors that receive these signals. They're classified based on where they are located within the cell. And the first class is intracellular receptors, so named because they are located within the cell. They can be located within the cytoplasm, or even, as in this example, within the nucleus itself. The type of ligand that can activate an intracellular receptor is very specific. So these tend to be smaller nonpolar, things that can diffuse across the cell membrane directly into the cell, thereby interacting with the receptor and eliciting a response. Most receptors that we think of when we talk about cell signaling are actually membrane receptors or cell surface receptors. And these are located on the cell surface, on the plasma membrane. They are integral membrane proteins. They tend to have an extracellular domain that can bind to the ligand and some sort of intracellular domain that can change in response to ligand binding to let the intracellular components know, if you will, that a ligand is present and that a transduction cascade should occur. The ligands that interact with cell surface receptors tend to be large or polar or charged, things that cannot diffuse through the plasma membrane directly and therefore need to interact with something on the surface instead. So quick concept check. Pause here and fill in the blank. You can review your notes or you can do this from memory, but take a moment to consider the following term. A signaling molecule is considered the blank. It's typically the smaller molecule that binds to a larger receptor. Okay, so this molecule is called a ligand, and that's what binds to the receptor and activates it. Second review question. Which of the following signaling molecules travel the furthest? Autocrine signals, endocrine signals, paracrine signals, or intracellular signals? If you recall from the slide a few slides ago, endocrine signaling describes signaling where a signal is released from a cell and travels through some sort of vasculature like the bloodstream or xylem and phloem to reach its destination cell. So those can travel very long distances. One more concept check question while you're on a roll of answering questions before we move forward with more information. And this is more of an application question, so consider the details in the prompt before you choose an answer. Here's the scenario. A cell is responding to a signal for which the ligand is a large protein that contains large patches of positively charged amino acids. Where would you expect to find the receptor for this ligand? On the cell surface, on the inside surface of the cell membrane, in the cytoplasm of the cell, or in the cell's nucleus? Right, so this is an application question because this isn't something you can simply memorize. So we need to consider the nature of the ligand, right? We know that it's large, and we know that it has positively charged regions, right? So those two factors exclude it from diffusing into the cell. So we can't have a receptor in the nucleus, in the cytoplasm, or on the inner surface. That receptor needs to be a cell surface receptor, or A. So now that we've talked about the general components and some of the terminology, let's talk about the actual process of cell signaling. There are three major phases of cell signaling, as I mentioned previously. There's reception. This is where the ligand or the signaling molecule binds to and interacts with the receptor. There's transduction, and this is a step or series of steps that's of variable length. Right? Some transduction events are very direct, and some involve lots of intermediates. Right? But this is where the signal is transduced, or the form is converted into some sort of form that the cell can understand and respond to. And then we have some sort of response, right? Something the cell does that's a change in reaction to the presence of that ligand. So activating a specific receptor of a specific signal leads to some sort of response, and what mediates that response, we call that signal transduction. As you see here, signaling can initiate a cascade of protein-protein interactions. And during those interactions, the signal can be amplified, right? Or more of it can be made, even though there was only one initial signal. The cell can make more and more so that it responds more readily. It can be distributed, so that there are multiple responses, right? Signaling pathways can also converge so that multiple activated signaling processes are taken into account when a response occurs. But ultimately, what's going to happen is that the cell is going to change something about its function or even its structure. So signaling cascades can include many steps, and these are sequential chemical reactions that occur. So in this example, and again, this is just an example, you don't have to memorize this particular signaling cascade, but we have a signaling molecule, ligand, bound to its receptor. Notice this tight specific fit. Now that this receptor is bound to a signal, we call this the activated receptor. In this example, there are a series of relay molecules and activated enzymes that get activated in sequence, right? So first this happens, this activates this particular enzyme, which then activates a second enzyme. We see energy is spent in the form of ATP, right, which feeds back on a previous enzyme and regulates it. We have another protein that's activated in response, and ultimately we get a cellular response. So this is a type of example we call a phosphorylation cascade because it involves multiple transfers of phosphates from ATP to intermediates to other proteins, ultimately changing their structure. Remember, you know the structure of a phosphate. It's got that negative charge that can change protein folding, and therefore it changes their protein shape. That can change their function. And then we have this cellular response, and this could be growth, this could be metabolism, this could be breaking something down, it could be building a structure, it could be changing shape, it could be moving, it could be changing gene expression, all sorts of different possibilities. So let's look at some of those responses in response to environmental changes. Here's an ion channel. We've learned about ion channels previously. We know that these are can be gated, and we've talked about the fact that ion channels can be regulated as to whether they're opened or closed. We've also talked about how they are specific to the movement of a particular molecule down its concentration gradient. And one of the things that can open or close or regulate or gate ion channels is ligand binding. So here we have an example of an ion channel in skeletal muscle, and this ion channel is opened in the presence of acetylcholine. So you see when it's open, this ion can move down its concentration gradient into the cell, and the response in this cell type is muscle contraction. Again, this is just one example of what can happen when an ion channel opens and when a cell responds. So in this case, we have the signal binding to the receptor. The receptor also happens to be an ion channel, right? The result of that is ions enter the cell, and the response is muscle contraction. This is a very rapid response because it has very few intermediates, right? So the cell senses the balance change in ion concentration between the outside and the inside. It changes the electrical potential of the membrane, and the muscle contracts. So that is a very rapid response. There are also sort of rapid responses, right? So here's another example. Here's where the signal itself is epinephrine. We've all heard of epinephrine. It has a specific receptor to it on the cell surface. And when bound and activated, the epinephrine receptor activates this enzyme called adenylyl cyclase. Adenylyl cyclase builds this molecule called cyclic AMP out of ATP. So the cell has lots of ATP in it. Adenylyl cyclase uses ATP as a substrate to make cyclic AMP as a product. And cyclic AMP in the cell is going to do a couple of things. One thing it can do is activate this enzyme called glycogen phosphorylase. And remember, we practiced figuring out what enzymes do based on their name. So we know it's an enzyme ACE. It's an enzyme that phosphorylates something. What do we think it phosphorylates? Glycogen. All right, so the end result here is that it breaks down glycogen into glucose monomers. Also, cyclic AMP can bind a different protein, glycogen synthase, inactivate it, and prevent more glycogen from being made. And these two outcomes both work to increase the glucose concentration within the cell, which can be spent as energy. So what's most important here? Not that you memorize what adenylyl cyclase does or what glycogen synthase does, but that you look at this and say signal, reception, transduction, response. We also have a little bit of distribution here, right? The same product, first of all, we've got amplification because the cell is making multiple cyclic AMP molecules from a single epinephrine binding event. And then we also have distribution that cyclic AMP is able to do two different things, in this case, both of which cause the same response, right, which is breaking down glycogen into glucose and making energy available for the cell. Also, it's a relatively rapid response because we're regulating the function of enzymes that are already there. They've already been transcribed and translated. We're just changing what they do a little bit. And there's very few intermediates between the signal itself and the response. Whereas altering gene expression is a much slower response, right? So gene expression can be changed. We've talked about the fact that first of all, gene expression regulation can be changed at lots of different points: before transcription, during processing, before translation, during translation, after translation, folding, localization within the cell, protein stability, all are points of possible gene expression regulation. And they all take time. If we look at regulation of transcription itself, so producing new mRNA transcripts that are then able to be spliced and processed and translated ultimately into new protein, which has a function, that takes some time. Transcription isn't immediate, translation isn't immediate. It takes some time to get from information in the DNA to functional protein. How does the cell know when and where to use the information in the DNA to build proteins? How does it know when to transcribe and translate particular genes? We talked about this generically by the presence of transcription factors. How does a cell know when a transcription factor should initiate transcription? In response to cell signaling. So let's look at this example here. In this case, we have a growth factor and its receptor on the cell surface. It changes in response to the presence of the growth factor and activates. We've already seen a phosphorylation cascade, right? So we've got some proteins that are already there that are going to get phosphorylated in response. And ultimately, what's going to happen in this example is we have the phosphorylation of a protein that's going to act as a transcription factor. Right? So this protein here, when phosphorylated, is going to do two things. It's going to travel to the nucleus and activate a protein that resides within the nucleus that, when activated, binds to and initiates transcription. So it's going to interact with the promoter region or an activator region on the DNA and recruit RNA polymerase and enhance the amount of transcription that's occurring of this particular gene. You see the mRNA transcript that's been produced. It's going to be transported out of the nucleus in this eukaryotic cell. And then we get the second function of this activated protein, which serves to ramp up the amount of translation of certain mRNAs, including this one here, which causes increased production of the protein, which ultimately is going to allow the cell to grow and divide, which makes sense, right? This is a growth factor, so the response is probably going to be growth. In order to get growth, we need cell division. This is a much slower response than either of the previous two we learned about because A, it's a couple different steps, and then B, we have to actually synthesize new protein. Right? So we're transcribing new mRNA. That mRNA is getting spliced and transported and translated. Those proteins are being folded, localized, and then they can function. So it takes some time. Also, there are more points where things have to be regulated properly for this response to occur appropriately. But the end result has to do with alteration in gene expression, which can change cell function. So at this point, we've talked about reception, we've talked about transduction, and we've talked about response. But the cell also has to regulate signal transduction because there has to be an end. Right? The cell just can't keep on responding forever. And you know this anecdotally, right? You don't want your neuron to keep firing a pain signal long after the pain stimulus is gone. You know it's there. You feel the pain, right? The cell doesn't produce proteins needed for cell division indefinitely forever the second it sees one growth factor and responds to it. Right? There's a point at which the cell stops getting the signal to grow and it needs to stop growing. So signal transduction also needs to have a way to turn off. One way is to convert the enzymes that are activated by the signaling pathway back into their inactive forms so that they are no longer signaling. It also allows them to then be re-stimulated when another signal comes along and is bound to the receptor. Right, so here we see an example of just one of the enzymes that was activated by a signaling pathway. Here's its active form on the right. In this example, it's got a phosphate that it got from ATP. But there's an enzyme that can then cleave that phosphate back off. It reverts back to its inactive state, and now it's ready to be activated again should another signal come along. Right. Here we've got a different example. We've got a protein that's inactive when it's bound to one nucleotide, a guanine nucleotide diphosphate, right? When there's a signal present, it strips off that GDP molecule and allows a GTP to bind, in which case it's active. And then there's an enzyme that cuts off that third phosphate, reverts it back to GDP. So again, it's in the off or inactive state so that it can be reactivated if another receptor binds to another ligand. Here's a third example. Remember we talked about cyclic AMP as a signaling molecule that can be produced in large amounts in response to a signaling pathway. Right, so we learned about an enzyme that can convert ATP into cyclic AMP, and we saw several different things that cyclic AMP can do for the cell. So how do we stop responding? Well, one way is to turn off the enzyme that made it, but then we've got a bunch of the signaling molecule, so the cell might continue responding. So we can activate a different enzyme that's going to break that down. Right, so once cyclic AMP is made, there's another enzyme that breaks it down into, in this case, AMP, which does not elicit the signal, and so the signaling turns off. So it's only when CAMP is present that the response is initiated. Once it's converted back into this form, we have no more signal until another signaling molecule binds to another receptor, in which case we activate this whole process again. So the key here is there is a balance between regulating the enzymes, and the enzymes determine what happens to the cell in response. So there are regulating enzymes and signaling enzymes, the signaling enzymes in that pathway that are activated, and then regulatory enzymes that turn them back off so that they can be reactivated later. And there are two major ways to do that, either activating or inhibiting the enzymes, or by controlling whether those enzymes are synthesized or broken down entirely. So some enzymes are actually degraded to keep them from signaling, and then they are resynthesized in the inactive form so that they can respond again. So I'm going to add one more thing to your things to remember, right? Reception, transduction, response, and then some sort of inactivation. Remember that all signals, in order to change the cell, need to have a way to be turned off so that that cell has a way to then respond again. We don't want cells that can respond once in their lifetime and that's it. It needs to be a cycle. We could have talked about cell signaling all day long, all year long, for years on end. Right? There are lots of cool applications. I teach a whole class on it. But the goal here is to think about, first of all, big picture. You've got a cell. It's enclosed within a plasma membrane. Only certain things can get in and certain things can get out. But the cell needs to know what's going on around it, what its neighbors are doing, what the environment is like, whether there are any stresses, whether there's food nearby. So how does it find out? Ligands and receptors. Receptors can be within the cell or they can be on the cell surface. How do you tell the difference? Look at the ligand and its properties. Can it diffuse into the cell or does it stay outside, in which case we need a receptor on the surface? When the signal binds to its receptor, the cell needs to notice that. There are one or more transduction steps, and then there's some sort of response. We saw examples where response was ions flooding in and muscle contraction. We saw examples where the response was regulating an enzyme's function or a protein's function. We saw examples where the response was changing gene expression, synthesizing entirely new proteins. And then all signals need to be able to be turned off.